Hi, my name is Zoe Drew, and I'll be going through my proposed research study and poster. So my proposed study is exploring the potential roles of oxytocin and glutamate in ameliorating the negative symptoms of schizophrenia. Schizophrenia is a neuropsychiatric disorder characterized by significant impairment to cognition, behavior, and emotions. Symptoms are typically categorized into positive and negative symptoms, where negative symptoms refer to behaviors and functions that are lessened or absent in affected individuals, and these are the focus of today's study. Despite the prevalent disease burden, the etiology of schizophrenia is not well understood. Historically, most literature has centered around dopamine, but antipsychotics targeting dopamine have failed to ameliorate the negative symptoms, and this has led to investigation into other neurotargets. Oxytocin, in particular, has garnered interest mostly due to its well-established role in social behavior, and also because it has exhibited some evidence of neuroprotective effects, and neurodegeneration is implicated in the pathophysiology of the disease. So there have been preliminary findings suggesting that intranasal administration of oxytocin can alleviate the negative symptoms of schizophrenia, but the mechanisms of this are unknown. Glutamate has also become increasingly prevalent in the schizophrenia literature as more evidence emerges that it's contributing to the physiology of the disease and specifically to the negative symptoms. The most prominent evidence of this is that the antagonism of the glutamate NMDA receptors can induce a syndrome of behaviors that mimic the negative symptoms of the disease. Also, inhibiting an NMDA co-agonist can induce structural neuronal changes that are characteristic of schizophrenia, such as reduced dendritic spine density and reduced hippocampal volume. So why am I discussing these two substances? Well, as mentioned, there's a gap in the literature as to how that administration of oxytocin is seemingly able to alleviate the negative symptoms of schizophrenia. And interestingly, we know that antagonizing glutamate signaling can induce those same negative symptoms. So putting two and two together, some people have inferred that or theorized that oxytocin's mechanism of action involves some modulation of glutamate. And specifically, some suggest that it works by counteracting glutamate hypofunction. And that leads me to my research question today, which is, does oxytocin ameliorate the negative symptoms of schizophrenia by counteracting glutamate hypofunction? The aims of this study are to investigate the behavioral and neural effects of NMDA antagonism or glutamate hypofunction in mice, to determine if oxytocin mediates this effect, and specifically to determine if oxytocin can preserve glutamatergic signaling. This study would employ a between subjects design. There would be 30 mature age male CD1 mice randomly allocated to 3 experimental groups. CD1 mice are a common laboratory strain of mice often used to model schizophrenia, and the use of male mice is to better control for oxytocin, as this varies less across males and also isn't varied by the estrous cycle. The 3 groups are the control group, the MK801 group, and the MK801 plus oxytocin group. MK801 is a non-competitive NMDA antagonist that's often used to model the negative symptoms of schizophrenia in rodents, and also at a neural level induces that glutamatergic hypofunction. So to make clear what the variables are, the independent variable is the treatment condition. So either saline control, MK801, or MK801 and oxytocin. And there are 2 dependent variables. So there's sociability, which will be measured by Crowley's 3-chamber sociability test. The exact steps of this will be explained soon, but the measure will be the duration spent in a sociable chamber as opposed to a less sociable chamber. The other dependent variable is glutamatergic activity, and this will be measured using whole-cell patch clamp recordings, and this will be done ex vivo on prefrontal cortex brain slices. And the measure will be the amplitude of EPSPs and the frequency of spontaneous EPSPs. And just as a side note, the nature of patch clamp recordings is why we have to restrict the electrophysiological recordings to one brain region, just for practical reasons. But the rationale behind using the prefrontal cortex is that it's been implicated in social behavior, and also there's functional homology between the rodent prefrontal cortex and the human prefrontal cortex. As for the hypotheses for this study, we hypothesized that oxytocin will ameliorate the negative symptoms of schizophrenia and preserve/restore the function of glutamatergic synapses. So the amelioration of negative symptoms is something we definitely expect to see. It's something that has effectively already been measured before or been observed before, whereas the preservation of the glutamatergic synapses has not actually been measured before. So this is much more of an educated guess, but is what is the point of interest of this study. And now just to go into the actual procedure. So first we have to set up the experimental design. So we select and obtain the 30 CD1 mice. We randomly divide the mice into 3 experimental groups. Again, the control, MK801 and MK801 plus oxytocin. We house them under standard conditions. Also calculate the appropriate dosages of MK801 and oxytocin. So once this is all set up, you undergo the induction of schizophrenia-like symptoms. This is via an intraperitoneal injection of MK801 into the MK801 group and MK801 plus oxytocin group, whilst saline gets administered to the control group. And then immediately after this, you have oxytocin administration. This again is administered by intraperitoneal injection, and this of course only goes to the MK801 plus oxytocin group. The other 2 groups get saline. After 20 minutes after drug administration, we can commence Crowley's 3-chamber sociability test. So here is a schematic of the apparatus used in this task. So it goes like this. The subject mouse is placed in the middle chamber. An unfamiliar mouse is placed in one side chamber, and the other side chamber is left empty. You then remove the walls between the chambers and allow the mouse free access to both, and essentially just record the duration that the subject mouse spends in each chamber. And this will run for 10 minutes. All right, so after the behavioral assessment, we go into the electrophysiology. So we have to sacrifice the mice so we can then prepare ex vivo brain slices of the prefrontal cortex. And then you just set up the patch clamp recording configuration, so such that you can be stimulating glutamatergic presynaptic neurons and then recording the glutamate-mediated EPSP on the postsynaptic neuron. So these are the hypothesized results from Crowley's sociability test shown in figure 2. So the things to note are that the MK801 group show, as expected, a preference for the empty chamber. So they prefer that non-sociable chamber, indicating their social withdrawal. And what's interesting is that the MK801 plus oxytocin group don't actually exhibit this same effect. So even though they've also had the MK801, the oxytocin is seemingly counteracting this effect on social behavior. So more pertinently, here are our patch clamp recording results. So as expected, the MK801 group is exhibiting the reduced EPSP amplitude and frequencies, indicating their disrupted glutamatergic signaling. But interestingly, this isn't observed in the MK801 and oxytocin group. Rather, their EPSP properties are pretty comparable to the control group, again indicating oxytocin is having an ameliorating effect. So what do these results tell us? These results support the hypothesis that oxytocin has a modulatory and seemingly protective effect on glutamate hypofunction in the context of schizophrenia. The results of the behavioral assessments indicate that MK801 did indeed reduce sociability in the mice, but that this effect did not occur in the mice also given oxytocin. The mechanism by which oxytocin alleviates negative symptoms is likely complex and multifactorial. However, as per the aim of the study, the electrophysiological measurements elucidated one more detail about this mechanism. Specifically, the patch clamp recording showed that glutamate signaling, which is typically dysfunctional in schizophrenia, can be restored by oxytocin. So what are the implications of this? Most prominently, these results provide insight, again, into the mechanism by which oxytocin may alleviate schizophrenia negative symptoms, something which is currently poorly understood. The results would illuminate the potential role of glutamate and oxytocin in the schizophrenia etiology, as well as their interaction with one another. And also, this reinforces the relevance of the prefrontal cortex, as this is where the analyses took place. More broadly, the findings would support the existence of neuroprotective properties in oxytocin and also implicate glutamate in social behavior. On top of the research implications, the study has clinical relevance, as it supports the growing body of evidence that oxytocin has therapeutic potential for schizophrenia. It may even support targeting glutamate more directly as a therapeutic option. It's necessary to acknowledge that the hypothesized results are not inevitable, particularly the hypothesized effects of oxytocin on glutamatergic synapses, as there is only moderate evidence supporting this theory. Even if the hypothesized results are observed, there is still need for caution about reaching conclusion and making inferences given the limitations of the study. There are a number of confounding variables other than oxytocin administration that could account for the preserved glutamatergic transmission. Additionally, the use of rodent models in schizophrenia is self-evidently not a perfect model of human schizophrenia, and there are often issues with how well these studies translate to humans. Also, the use of exclusively male mice makes this study less translatable to females, and inherently means the estrous cycle has not been accounted for. And finally, it's necessary to mention the study has a fundamental drawback, that it's highly based on inferences rather than establishing a clear causal relationship. In the statistical analyses, it may be beneficial to investigate that the behavioural task performance correlated with the electrophysiologic recordings. That is, ensuring that the mice who exhibited restored sociability with oxytocin administration also exhibited restored glutamate function. However, even if a strong correlation is observed, this is not sufficient to infer that oxytocin is the sole cause of glutamate preservation. Thank you for listening.